Hello everyone. Today I will demonstrate the analysis process of the water maze experiment using our software VisuTrack. Enter the password. First, create a new experiment file. Enter the file name MWM. Here you can see the file information, file name MWM, default storage path of the file. Default storage is under experiment in the software. Tracking mode is based on motion trajectory. If you are testing forced swimming, you can select the following based on activity. Click on the upper left experimental platform. You can see a real-time video. We are demonstrating an offline water maze experiment. So click on the right test and select the video we are demonstrating today from the file. As you can see, here is a video of a water maze. First, we need to draw an outline for the water maze. Generally, a circle is chosen. The size of the circle should be flush with the maze. After alignment, we choose to add a grid, or sector. We choose to add a 90-degree sector. As you can see, the water maze is drawn into 4 sectors. If the camera is not aligned and the partition is inaccurate, what should we do? We can choose the 45-degree grid here. Then select the inappropriate line and click Delete. Sometimes this happens. We need to make partitions according to the actual situation. Next, we will calibrate the camera scale. First, draw the diameter of the water maze. Generally, when doing rat testing, the diameter of our water maze is 1.6 meters, or 1600 millimeters. After setting it up, put the scale aside. Next, we add a hypothetical hidden platform to the water maze. Add it here. Once the platform is added, we create the regions. Add 4 subregions to the 4 sectors. Add a general tracking region for the hidden platform. Select the subregions and select the corresponding sector. Select the general platform and select the small circle. The next thing we need to do is to identify rats. First, click on the identification settings on the left, then click on Smart Identification on the right. Using the box, select Rats. The current recognition algorithm is center of gravity recognition. If we want to add a plug-in, we need to select 3-point recognition. The blue point represents the head, and the green point represents the tail. After the recognition is set up, we need to set a test parameter. We usually test the water maze for 90 seconds. That is, after starting the analysis will stop after 90 seconds. The head exploration behavior and state switching are not used much in the water maze. If you need it, please contact us for more information. The last option on the left side of the experimental platform setting is the plug-in selection. We provide a plug-in specifically for the water maze. Click Enable Plug-in here, and then select the platform area. The platform area we just set up is this small circle. There will be an animal perspective looking towards the platform. This is a 30-degree sector for each eye, for a total of 60 degrees. Open the real-time water maze plugin. We can see that there are several parameters here. The first one, the linear search, is based on the central axis of our animal's entry point and the platform, with a width of 15. If the rat searches in this area, we consider the rat's search strategy to be a linear search. Marginal search means that we define an area. If the rat is in this area for a certain period of time, we think that the rat is conducting a marginal search. Look here. Sometimes we see the rat's movement track is sometimes white, sometimes red. Why is that? Look at this fan-shaped perspective. When the line connecting the rat's head and the center of gravity is within this fan-shaped, we think it is moving towards the platform. If the connecting line is not within the fan-shaped, we think it is moving away from the platform. When moving away from the platform, this line is white, and when moving towards the platform, this line is red. Next, let's set up a simple group. For example, Group A has 10 rats. In the acquisition analysis, you can see that 10 tracks have been generated. Let's select a track. If you are using real-time video, just click the small eye in the video option. We are currently demonstrating an offline video. Open the folder and select a video file, then click on the video to play. The video has not yet started to be analyzed. Click Start Analysis. The analysis time is 90 seconds, which is what we have set before. This is an offline video, and we can play it at a maximum speed of 8 times. Real-time video cannot be accelerated. We can right-click on the video to open the behavior record table and behavior event graph. There is also a real-time plugin for the water maze, which can observe the movement of the mouse, including the distance from the platform and the angle from the platform. The software will count these data. After the analysis is completed, the video position will generate the activity trajectory of the rat. This trajectory diagram can be viewed in the data visualization. Some of these trajectories are green and some are red. As we just said, red represents the rat's tendency to move toward the platform. If you want all the track lines to be the same color, you can click on the right side, Track Chart Parameters. Click the 2 arrows of the center of gravity track color. Change the color of the approaching platform and the away from the platform to the same color. This will give you a trajectory map of the same color. You can also use the track image parameters to hide the entire circle platform. Click the second option below to see the heat map parameters. Heat maps are of little significance in the water maze, so we won't introduce them in detail. The third option below is the behavioral event record sheet. The behavior software of the entire experimental rat will record when each behavior started. The fourth option below is a data visualization record table. The time axis here is 60 seconds. We can change the 2 on the x-axis to 3 to display 90 seconds. If the movement speed exceeds the upper limit of the y-axis, we can also modify the y-axis. Finally, here are some data charts. The software displays the access time chart by default. We can drop down and select the data chart we want to display as needed. We can export the image, usually to Data, in the experimental folder. Here is the data visualization table. The main experimental data of this test has been generated here, such as the time the rats spend in each region, the number and time of exploratory behavior, etc. If we want to add data parameters, we can right-click on the chart and check the parameters of interest. We can also view the data in segments, such as between 10 seconds and 70 seconds. Here we show the data for 60 seconds. Sometimes the rats will enter a diving state, which the camera cannot capture, so the time here will be different. Finally, this data can be exported into a table. This is the process of analyzing the water maze using our software VisuTrack. If you have any questions, please contact us.